Hello and welcome back to our course, Data Science for Drug Discovery, Health, and Translational Medicine. In this lesson, we look at bioinformatics and how it applies to medical science and medicine. In the previous module, we learned about bioinformatics and something about the central dogma of molecular biology and how bioinformatics came to its current state. But we really didn't learn specifically how we could use bioinformatics to improve human health and learn more about medical science. And in that sense, there's an analogy to the state of things in about 2000. And so this slide is about how at the end of the Human Genome Project, there was a lot of hype. As Francis Collins said, then what more powerful form of study of mankind could there be than to read our own instruction book? And in order to generate the $3 billion in funding, there were some suggestions by scientists and politicians perhaps that this project would provide faster results than was the case. So there was a lot of hype and headlines and magazine covers like you see here. And I've also put on this slide this Gartner's hype cycle, which I think applies to this situation quite well. And notice the steep rise in expectations is called the technology trigger. Then you reach a peak of inflated expectations. Then there's a trough of disillusionment, followed by a slope of enlightenment and a plateau of productivity. I am not sure where we are on that upslope, but I can tell you that there was a peak of inflated expectations and some disillusionment. Yet I think most scientists in the field today would agree that the Human Genome Project was hugely successful, productive, and led to so much advance in science and in medicine. This module is about bioinformatics and medicine, how bioinformatics can improve medical science and practice. And so let's get into the subject by looking at an example, a very nice example, and it is the drug Gleevec. Gleevec, as it says here, was a breakthrough in cancer treatment, specifically a leukemia treatment, and specifically a form of leukemia called myelogenous leukemia. The Gleevec story is not all about bioinformatics, and it goes back several decades, but much about the story and how Gleevec was discovered and why it is a great success is related to bioinformatics and genomics, and so it really is suggestive and encouraging. The drug Gleevec inhibits a tyrosine kinase, a protein involved in cell division, or mitosis. Acute myelogenous leukemia is AML and associated with a chromosomal abnormality in leukocytes, or white blood cells. This was discovered in the 1950s through a relatively new area, cytogenetics, or the genetics of cells. Not organisms, whole organisms, but the genetics of individual cells, as they realized that cancer cells had their own genetics, their own genomes, different from other cells. So these abnormalities in the cancer cells are a somatic mutation involving the BCR and ABL genes. And that was elucidated in the 1980s through more advanced technologies such as sequencing. The mutated ABL gene is an oncogene, meaning related to cancergenesis. And the two genes, BCR plus ABL together, are expressed in the abnormal tyrosine kinase, which causes or is associated with AML. The discovery in the 50s of this chromosomal abnormality through cytogenetics is illustrated by the left diagram here, or photo, which illustrates the so-called Philadelphia chromosome, named after where it was discovered, the lab. And on the right, we have the ABL1 protein bound to Gleevec in its binding site. which is illustrative of the success of Gleevec as this anti-cancer drug. It really was something of a wonder drug, and I'm going to read from this source, which I highly recommend, from Nature Education. In 1998, this chemical, which was called ST1571 at Ciba-Geigy, and eventually renamed Gleevec, entered its first clinical trial. Small phase 1 trial involving just 31 patients. Remarkably, all 31 individuals experienced complete remission. In other words, their blood counts returned to normal. In some of these patients, there was also cytogenetic remission, meaning that the Philadelphia chromosome was no longer found in their blood cells. As Drucker, the scientist, noted, that was virtually unheard of in a phase 1 clinical trial. Usually in a phase 1 clinical trial, if you see 20% response rate, that's remarkable. We had a drug that was extremely well tolerated and had a 100% response rate. It was incredible to see this unfold. So for the cancer patients who have benefited from this drug, it truly has been a therapeutic marvel. In telling that story of Gleevec, we use some terms which may not be familiar. One in particular I'd like to point out is the idea of a somatic mutation. And the distinction to be made is between germline and somatic mutations. Germline means the DNA which is passed from parent organism to offspring. Somatic refers to the body and not the germline, so a mutation in a particular cell, such as a skin cell or a cancer cell. And so, as it says here, genes and chromosomes can mutate in either somatic or germinal tissue. These changes are called somatic mutations and germinal mutations, respectively. There is a particular relationship between cancer and cancers and genomics and bioinformatics, and I'd like to emphasize that as indicated by the Gleevec example and also through this recent paper, 2010, all cancers carry somatic mutations. In many ways, cancer is a disease of somatic mutations where specific tissues become different, become mutated, and their behavior becomes that of cancer. For the remaining portion of this lesson, I'll be making good use of an online resource called SciTable from the Nature Publishing Group, NPG, and one of their efforts called Nature Education. So it's highly recommended. We'll be showing a little bit from that resource, but I encourage you to look further into that SciTable resource on genes and disease. The hereditary nature of some diseases has been known for some time and certainly predates the modern molecular understanding of genetics and genomics. Yet, with bioinformatics and modern genomics, we can understand hereditary birth defects much better, how they might be treated and understood and prevented. Huntington's disease is a great example of a hereditary disease which was understood on some level long ago, but with improvements in technology and our understandings of molecular biology, bioinformatics, and genomics, our chances of addressing the disease at a molecular level and treating it have gotten better and better. From that same collection of lessons from Nature, this is a good one, again, relating bioinformatics and genomics to cancer and understanding what we mean by oncogenes and proto-oncogenes, meaning genes which can become oncogenes and thereby cancer initiators. This screenshot is from another online course called Code of Life When Computer Science Meets Genetics, given by the Hasso-Plattner Institute of Potsdam, University of Potsdam in Germany. And it reflects how genomics can be brought to the clinic, meaning to a health provider, a physician trying to treat an individual patient. So here, this fictional patient has certain biomarkers, meaning certain genomic patterns, and in this hypothetical situation, a doctor would be asking what sorts of treatments would be most appropriate for this patient given their genomic patterns. So that would be considered a form of genomic personalized medicine. I encourage you to consider that course and some of the resources provided by that institute. As always in this course, we're not trying to cover everything, which would be impossible, but mostly to consider how we as data scientists can be of use, can bring our tool set to these areas and assist, whether it's in the more basic research areas or to patients or individuals. So let's go through some of those ways relative to bioinformatics and medicine. One is simply what I call bibliology or library science, providing tools which can access the vast amount of literature and data online, whether it's relative to a particular disease or a particular organism. And I also put together with that World Wide Web programming, the ability to harness online APIs and other resources to bring the appropriate information to bear on a problem. A particular kind of analysis is called GWAS, or Genome-Wide Association Studies. And typically what we're doing is sequencing an entire genome of an individual or a cell and looking for the single nucleotide polymorphisms, SNPs, and trying to associate those with some phenotype, typically comparing a diseased tissue with a normal tissue. Gene expression data analyses refer to the differential expression of gene products, normally proteins, in varying tissues. So it may be that particular genes are expressed more in one tissue than another. That can give us clues as to what's happening at a molecular level in diseased tissue. However, I'm putting that bullet there, present versus functional, because there are big questions as to whether gene expression is simply being present or actually doing something functional. Sequence and alignment and similarity analyses, for example, BLAST. This is a big area of bioinformatics. Can we compare protein sequences and infer relationships, whether common ancestor or functional relationships between different proteins? And that applies to the next point, cross-species analysis. It may be that we have more data on a different organism, but by comparing sequence, we can then associate with the organism of interest, typically Homo sapien. These associations, whether via similarity or in functional relationships, together comprise what we might call a gene-gene or protein-protein interaction network. And network analysis is a form of, or a branch of computer science and analytics which data scientists can bring to bear. Last, pathways, reactomics, metabolomics, again refer to many of the interacting networks, biological networks, which comprise physiology and pathology. So what are our take-home messages today, to sum up? Bioinformatics addresses the molecular basis of health and disease, so it must be important. Genomic variation between individuals, tissues, and cells can help explain and address disease. So it's one thing to have the genetic code. It's another thing to recognize what are the variations which makes someone sick in a particular way, or makes one cell or tissue diseased versus another, even within one individual. Bioinformatics and data science can be used across the translational spectrum, meaning from basic science to clinical science, and in medical practice, and even in personal health. In the assignment, you'll be using some data science tools to address a medical question using bioinformatics. Until next time.